Good afternoon, everybody. Let me thank you for the opportunity to share with you our thinking about value sharing of genetic resources and information. I'll start with a brief description of our organization. So our mission is active support and effective use of natural resources in equitable manner. Our main business is genome profiling service and IT support for agriculture and ecology. We're operating since 2001, so we just turned 18 a few months ago, and we were first affordable whole genome profiling service. We are now a global service provider with 2 million whole genome profiles and over 1,000 organisms delivered to users from over 60 countries. Our support for international agriculture is mostly through technology development, but also through training and services. We also collaborate in technology package delivery to other countries, with Mexico and Africa our first locations. We have currently a team of 45 that includes a lab, data analytics and IT people, but plus international visitors and trainees. We have high diversity of clients, from individual farmers to multinational corporations, but the majority of our users come from small to medium-sized enterprise and public institutions, including most CGIAR centers. Most of our clients are involved in genetic improvement and characterization of genetic resources. We have this enormous diversity of clients that represent equal diversity of needs, therefore require diversity of solutions, and over the last 18 years we work towards this goal. We are a bit unusual organization. Why our company has proprietary in the name. We are somewhere between social enterprise and B Corporation, for those of you who are familiar with B Corporation concept. B does not mean second class, and I don't have time to explain this any further. We're definitely a very non-traditional company. We have no profit focus, no investors, no formal board, no sales staff, and no lawyers. We have an ethnically diverse team with a mixture of skills covering molecular biology, chemistry, electronics, mathematics, and stats, and information technologies. We are organized into 3 main domains of operations with significant overlap and also migration of staff across these domains. We're operating in a chaotic manner, and again, it's a teaser. I don't have time to explain what chaotic really means, but we really have family-like atmosphere in the company. We also do a lot of training at DART, not only for our own staff, but for over 200 trainees since our inception. Most of our trainees come for just around 2 weeks. Very often will bring their own materials that we process and develop a good understanding of a technology and work with us on downstream data analysis. We had also over 20 long-term trainees who spent either several months or over a year, mostly in the context of technology transfer to other institutional locations, and we embed in them full competence of delivering technology package. DNA technologies on offer at DART can be grouped into 2 categories: whole genome profiling, with over 20 years in technology development of complexity-reduced representations. The most familiar technology for many of you is DART-seq, which is very highly ranked genotyping by sequencing technology. There is also its cheaper version, DART-seq LD. We work also with methylation, and more recently on resequencing using sequencing of low copy fraction of genomes. All these technologies have high data quality and extensive quality control embedded in the whole process. But we also have targeted genotyping methods. We have developed several of these technologies from 2014. DART-MP, DART-TAG, which is molecular padlock-based genotyping, and also DART-CAP, which is RNA capture probes-based genotyping of selected DART-seq markers. We choose appropriate technology to appropriate task, and also, since all these technologies are highly scalable, we define and use appropriate scale of the technology to provide solution to a specific problem. There is a lot of applications that we've done over the years using our technologies package, from comprehensive characterization of germplasm and diversity studies. Many of those studies involve gene bank collections, characterization, and most CGIAR centers had their genetic resources characterized on our platforms. We do a lot of genetic identification in the context of PBR, but also in determining seed purity and quality. We work in Australia, but also in Africa and Asia with both individual farmers and bulk handlers, but also we've done a lot of studies on varietal adoption supported by Bill and Melinda Gates Foundation, World Bank, and FAO. We've done over 1,000 mapping populations and a similar number of GWAS association mapping analysis, build also consensus maps for several crops as a community service. The bulk of our work in agriculture is in the field of genomic selections, both in plants and animals. But we also have a significant engagement in ecological research, in population genetics, studying invasive or endangered species, monitoring global fisheries, working in applied ecology, in biodiversity conservation, and also in genetic identification of organisms. Within the company, we have two domains in information technology area. The one on the left is dealing with the DNA data production. The one on the right is data integration domain. And those two domains are now fully integrated and operate as a package. Our data integration system is based on a KD-DART platform. It is built on open source tools, both our full data model and complete API, and most of the applications are either open source or free to use. I don't have enough time for a full description of the KD-DART platform, but I will run for you a very brief section of the informational video that we prepared for the platform, focusing on a couple of the applications from the suite which were recently modified for AgroFIMS project within the big data platform, and those two applications are KD Smart and KD Explore.
KD Smart is an Android application designed for scoring data for field trials and nurseries. It's available for free on the Google Play Store. Breeders usually measure traits such as plant height or performance in field trials by scoring and storing data in KD Smart. Trials can be created within KD Smart or from data in a CSV, or transferred from other parts of the KD-DART suite in a proprietary KDX file. With plenty of scoring options available, users can collect data in the way that suits their needs, minimizing time required while maximizing data quality. Still images and audio recordings can also be collected as data through the app. KD Smart has the ability to scan barcodes to navigate to plots and score traits. When scoring has been finished, scored trials can easily be exported to KD Explore for data curation and exploration, or exported as a CSV file for use with legacy systems. KD Explore is a desktop Java framework for plugins developed either by Dart or third parties. These plugins have a focus on applications for plant breeding, genetics, and biological sciences. It can be connected to a KD-DART database or used as a standalone offline application. The KD Explore plugins provided by Dart deal with trial management, trial design, data curation, and visualization of data. Trial Manager is the KD Explore plugin that enables users to create trials and manage trial data. Data that has been scored with one or more KD Smart devices can be seamlessly and securely transferred to KD Explore for data curation and visualization. Once data is curated, it can be loaded into a KD Dart database either directly or via KD Manage.
Let me finish then with our perspective on benefit sharing issue. We focused for 18 years on technology development and delivery both in genetics, analytics and IT, but technology itself is not enough and sometimes it brings opposite effects. Some of you may have heard the story about the inventor of the plastic bags who invented this technology to protect trees. Unfortunately, his invention clearly terribly backfired. Technology distribution is through open access and open source licensing, but also a lot of training and partnership. We're enabling local capacity for solving local problems. Ultimately, we need to go beyond science and technology and not only communicate better with intended beneficiaries, but also deliver more tangible outcomes. Most of the technological innovation is still primarily benefiting those with resources and power, and we need to find more equitable sharing arrangements. And ultimately, we'll need to change our value systems, but we are starting now with creating incentives for a better behavior. That's all what I have to say. So thank you for your attention, and hopefully you have interesting discussions. Goodbye.